work from the lab regarding the malignant B cells and therapeutic T cell treatment. I guess in the immune disorders in both animal models and in human patients. So without further ado, thank you so much, Gang, being with us today, and we are looking forward to your talk.
Sure. Thank you, Dr. Wu. Thank you very much for your nice and very detailed introduction. And this is really my great honor to be here to share with you what these most updated research progress in our lab. So let's just get started, I think. Okay, so today I'm going to talk about mainly two stories recently have been developed in our lab. One is about the how we delineate the very particular metabolic vulnerability of malignant B cells, which mainly which is mainly about the B cell acute lymphoblastic leukemia and the other type of the B cell tumors like lymphoma. And also, I would like to also spend most of the time to introduce our most recent work to find one of the very unique metabolic pathways called mevalonate, so which you guys could be familiar with, which is very important to produce cholesterol in the cell. And what is the role of this mevalonate pathway activity in regulating the T cell differentiation during these anti-tumor immune response, which we mainly using the human derived CAR T cell as the model. So I would just start with a few slides with the background. So this is mainly the scope of our lab study. So we are very interested in find out the mechanisms of the metabolic regulation, which is a temporary and a special reprogramming of the metabolism that will govern the malignant progression, as Dr. Wu just mentioned, the initiation, transformation, and progression of the B cell malignancies, including leukemia and lymphoma. And also will be another interesting part will be to study how the anti-tumor T cells or therapeutic T cells got exhausted, or how the fate decision mechanisms, which will be related to metabolic regulation in these two parallel direction, which I mean, it could be very rational to me because those two parts of the cells are mainly the very similar in original coming from the lymphoid progenitors. So when we talk about the metabolism or cancer metabolism, actually Otto Warburg's studies, which almost a century ago, which initiated a very interesting field, people found that actually under the even the very sufficient oxygen supply, the cancer cells would undergo glycolysis rather than oxidative phosphorylation. Of course, now we know it is not because of the defect of the mitochondria activities, but it's becoming a kind of the very unique choice the cancer cells make to use glycolysis as actually a way to produce more metabolic intermediates for the biosynthesis. And one very interesting phenomenon after several decades, which is observed by John Barker's lab, which they found when you stimulate T cells in vitro culture, you found actually the similar behavior of those T cells like the cancer cells observed by Otto Warburg, which you can see very fast consumption of these glucose after activated those T cells, and the accumulation of lactate in the culture medium, so which also later be identified as the aerobic glycolysis in T cell during the activation and the proliferation. So basically, after combining all those studies, we found that very interesting thing is these lymphocytes, including T cells and B cells. From morphological observation, we can see that most of us will be familiar with. Actually, the lymphocytes have very unique morphology with the very huge nuclear and a very limited cytoplasm. And then consistent with this morphological observation, actually, there is a very detailed comparison in terms of the B cells with the control, for instance, myeloid cells, you can see that once activated, the B cells or lymphocytes are proliferated much more faster, but they have actually the much more smaller cytoplasmic volumes, and more importantly, each cell contains way more less number of mitochondria in the cells, which indicating these B cells could have some kind of urgent needs for raising up the activity of the mitochondria in a very short time, which to meet the needs of the energetic or metabolic needs for proliferation. That's why once we check these AMP ATP ratios, you can see those B cells, once activated, they compared with the other type of the myeloid lineage cells, you can see they have kind of emergency in energy supply. There's kind of a deficit or make the kind of a limitation for those B cells to be fully capacity in proliferation. So that's why once based on this observation, we would like to test if during the malignant transformation of those B cells, they undergoes uncontrolled proliferation, not just like the normal activating process, which could be anyway be terminated, and the cells will goes back into the quiescent state. But with the malignant transformation, it's kind of a non-stoppable process, which those B cells are just to keep growing. And after studying these metabolic limitations, we found that very interesting. For instance, the PAX5 as the B cell unique transcription factors, they are not only regulating the development of these B cell or pre-B cells in the pro-B cells in the bone marrow, but they also performing like a gatekeeper for the metabolic restriction for those B cells. Once they are activated, the PAX5 starting to restrict the metabolic activities, including glucose uptake or other more activities, metabolic activities, to make sure those B cells are not proliferate in an uncontrollable way. So which we make it as we named it as a metabolic gatekeeper. So the idea is actually quite similar with what Dr. Chi-Hong has been proposed for T cell activation, right? He mentioned it as a metabolic checkpoint for the T cell. So once you activate it, you got signal 1, 2, 3, but you still need the metabolic adaptation for the T cell to really working together with all those signals, extracellular signals, to make sure the T cells can proliferate. So it looks like it's a similar phenomenon in B cells, and it's also controlling the malignant transformation. And our previous studies have been identified like the endogenous PAX5 and other transcription factors like EBF1, the B cell specific transcription factors, are actually controlling the endogenous activities of pentose phosphate pathway of those B cells to limit the production of NADPH, which is we define it as another endogenous metabolic gatekeeping pathways to prevent the cells produce too much NADPH, and then to generate those biosubstrates for nucleotide biosynthesis, all those ways. And then we find the very unique molecular mechanism, which is mediated by PP2A to control the dispense of the glucose between the glycolysis and the PPP pathway. And also very interestingly, as I previously mentioned during the discussion, the B cell malignant, they are actually inherited from these normal counterparts. Once these pre-B cells or pro-B cells have this unique metabolic regulatory machinery here, the malignant B cells usually inherited it from these normal counterparts, and then we find it will be a very unique metabolic vulnerability for us, maybe to really eliminate these malignant B cells during the progress of the disease. So this is our previous studies, and after establishing our own group, we are still very interested to see if there is more unique metabolic behaviors or mechanisms being identified in BLL, and also including the other B cell malignancies. And the reason why BLL is still a kind of interested to us is because after so many years development in the advancement in the clinical treatments, actually including the CAR T treatments, we can still see the adult BLL patients still experience high rates of relapse. Even after CAR T therapy, we see that like nearly half of the BLL patients will experience relapse after this magic or bullet-like treatment with the adaptive T cell transfer therapy. So actually, it's still quite a big issue or challenge to human. And also, we can see that once this patient got relapsed, the prognosis is really bad. So it's 5 years or survival will less than 10%. So lab members from our lab, postdoc Liu Huiming and the two PhD students Song Junzhe and Ji Xiaotian, they decided to study this most malignant subtype or to find out what is the metabolic trait between this difference between the normal and the most malignant or aggressive BLL subtypes. So from their studies, actually, they found a very interesting signature or the molecular signature, which is not strange to us, is like the MYC activity and the PI3K activity combined together will help to indicate a group of the patients if they have higher MYC activity combined with the PI3K activity, they always have a very bad prognosis. And once we compared the studies from the clinical cohort of the BLL patients compared with this newly diagnosed and the relapsed patients, we can find that these two signaling pathways or transcription activities are got highly activated or increased in those relapsed patients, which indicating, yes, these two molecular signatures will be very powerful to indicating the malignancy or the aggressiveness of those BLL tumors. And then after by collaborating with the physicians in Zhejiang universities, actually we collected our own sample group from the BLL patients, and we also here to show 10 patient samples. We divide them into newly diagnosed group and the relapsed group, and we identify like it's quite similar with these publicly available cohorts. What we found by Western blot, the MYC expression levels and the PI3K signaling levels are much more higher in the relapsed BLL patient. So they decide to study what is the metabolic behavior or what is the unique of the metabolism those very aggressive BLL cells have. So by doing this, we have to build up the mouse models to study. Otherwise, it will be not very feasible to perform all kinds of these assays. So by using the primary bone marrow derived pre-B cells from the donor mice and then transformed them with the NRAS G12D or BCR-ABL to generating the BLL cells, we can generating the mouse BLL cells from different background, genetic background of the donor mice. And you can see that we can find a inducible deletion of PTEN to including to inducing a PI3K high signature and overexpression of MYC, or you have both in the same BLL cells. And when comparing these signatures with the human samples, we find that those mice cells are nicely recapitulate the at least the transcriptional level signature of those human MYC high, PI3K high relapsed BLL cells. And also in the preclinical model, we see that very faithfully recapitulate the progression of the disease, which is basal BLL cells have the most prolonged survival, while those MYC high, PI3K high BLL cells have very, very bad prognosis compared with these control tumor cells. And then, of course, it will be very interesting because we know that both MYC and the PI3K are masked regulator of the cell metabolism, right? So here we can see that after by roughly measuring the glucose consumption and the lactate production, all those double mutant or the MYC high, PI3K high BLL cells have these highest consumption of glucose, and then the glycolytic activities have been shown here. Most importantly or strikingly, observation we have is by electron microscopy. We found that actually the double mutant or the MYC-high PI3K-high BLL cells have a very significant increase in number of the mitochondria and also the density of the Christie for each mitochondria we can observe here. And inconsistent with this morphological observation, we also see very high levels of OCAR, so which means those cells are undergoing very active electron transportation pathways to producing ATP in the cells compared with the other control counterparts. And we, when applying with the C13 labeled metabolic tracing experiment, we use two main nutrients which will fuel the TCA cycle. One is the glucose and one is the glutamine. You can see how these nutrients have been uptaken and being used up in the atom level in the TCA cycle. And after analyzing all those metabolite levels, we found a very interesting pattern, which you can see the oxaloacetate, which is OAA, indicated here in this, is actually have a much lower level in those MYC-high, PI3K-high BLL cells, and also the metabolic derivatives from this OAA acetate also have the lower level. But the next step, metabolite citrate actually got elevated. The levels got very high enrichment of the citrate levels in those cells, no matter the atom is coming from or these they are derived from the glucose or glutamine. So that means the cells are becoming a divergent metabolic regulation to really produce more citrate rather than generating in general more TCA metabolites. And after look at these lower how citrate has been used up, you can be used into these to generating the alpha ketoglutarate in the TCA cycle, or the or we take it as another way, like the ratio from product to precursor, right? It is always indicating how what is the efficiency of this metabolic process is. Once we calculated those ratio, we can see that the OAA from generated from the malate, the metabolic activities that got reduced in those cells, but the citrate generated from OAA got highly activated, which is very consistent to what we've observed in the amount of this metabolites. But then later, when alpha ketoglutarate generated from citrate levels also got highly repressed in this level, so which we found actually citrate will be a very unique divergent point for those double mutant BLL cells in terms of the TCA activity. So why the cells are doing this, and what is the consequence of this? So because in general we know those cells are very malignant, right? They are growing much more faster and more aggressive in vivo. So what is this observation has anything to do with this cellular behavior? And as I previously mentioned, actually one of the very important amino acids being generated from the TCA cycle is aspartate, and aspartate is actually produced from the oxaloacetate, which is quite repressed or decreased the levels in those double mutant cells. And once we compared with all those 20 amino acids in those 4 different type of tumors, we can see only aspartate levels got reduced in those MYC-high, PI3K-high cells rather than other type of the amino acids which are highly enriched to support the growth of those cells. And as I previously mentioned, one divergent point is the citrate, right? So as we know, the mitochondrial membrane is not permeable to all kinds of metabolites in the what happens in the mitochondrial matrix. But one of the materials will be transported out or in from this mitochondrial barrier, the membrane will be citrate. We know that, for instance, the acetyl-CoA cannot really be diffused out or transported out from the mitochondria into the cytoplasm. But one metabolite with the citrate very important is to produced in the mitochondria, and they will be transported out into the cytoplasm. And when measuring the levels of the citrate, which is this distance we previously observed, the overall the total citrate levels got highly activated. The cells are hoarding all those citrates in the cells, but not providing them enough to go downstream into the TCA cycle. And you can see that if we measure the mitochondrial citrate by separating the mitochondria out of the cells, we found the opposite observation, which the citrate levels in the mitochondria actually decrease. So, which this gives us the conclusion, like most of the citrate being produced or elevated, generated in the TCA cycle are transported out into the cytoplasm. So, as we know, the very, very important role of citrate being generated in the mitochondria will be transported into the cytoplasm, will be used to generating lipids as the precursor for de novo lipids generation. And this is also being observed or have been reported by other groups, which they found in the mammalian cells. All these metabolic process to generating or biosynthesis of, for instance, lipids and other type of these amino acids or nucleotides in the way of how many NAD they were need to use as a cofactor. So which you can see here, actually, no matter the lipids are generated from these carbon source of the glutamine or glucose, they are actually the most highest in the need in terms of these NAD as a cofactor to help the cells to really go through to generating the lipids. So this makes up to hypothesis like the highly generated or the highly elevated levels of the citrate in the cells could be reflecting some certain levels or the changes of these NAD level or NAD/NADH ratio in the cells to reflecting this redox status of the cells. So let's take a few more minutes to really recall what is actually the cofactor NADH or NAD as the electron donor or receptor happens, what's their role in the metabolism. So actually we know here on Earth, the planet, the life is actually originated coming from the energy coming from the sunlight, which is a very, very unique. We are lucky we have this very ordered energy have been continuously input into the transferred into Earth. And then we plant receiving this energy from sunlight. Then we use the water as the electron donor to extract out this low energy level electron from these molecules and using the carbon dioxide as the source of this material and as a nutrient, so which to fixing these low levels of energy to putting them, combine them with energy get from the sunlight to becoming a high level energy electron, and then generating glucose as a carrier of those high energy electrons and pass it to animal. At the same time, they're generating oxygen, right? So when the animals consuming those plants or glucose, more detailed in the cellular level, the glucose will go into the mammalian cells and go through as what we all familiar with the glycolysis and the TCA cycle. And then the high energy electron will be extracted and passed to NADH, which we know is a classic or most important function of the TCA cycle to generating high energy electron and pass this to ETC, electron transfer, electron transportation train to use it extracted energy from those electrons to generating ATP. At the same time, the oxygen will be used as the final acceptor of those low energy electrons, and then the cells will excrete out as a water as the final production, as the final product to carrying those low energy electron out of the cells. At the same time, you're generating ATP, you're generating the carbon dioxide, and they give it back into the environment. And the ATP will be used to support all, as we know, the biological process. So this is seemed to be a very perfect circulation or cycle of electron in terms of the life happens on Earth. But here is the problem. The mammalian cells, sometimes they need to change their activities of particular metabolic pathways. And by introducing this perturbation of this metabolic network, sometimes the electrons will be not so easily passed directly through to the ETC and then being generated into water. Sometimes you have to waste them because, as I previously mentioned, the cancer cells or activated lymphocytes, they have this activity called aerobic glycolysis. The most important phenomenon we observed is that they excrete a lot of lactate as a waste to proceed to make sure this glycolysis can keep going. And why this is particularly important to generating lactate is because when the high energy level of glucose cannot be used all the way down to the lowest level of the energy, they have to be wasted into lactate and be excreted out of the cells to make sure the whole process will keep going, but not by traffic jam, cause the traffic jam of those intermediate levels of electrons in the cell. That will cause problem to inhibit a lot of other metabolic activities. So this is a compromise which the cells or the life on Earth choose to adapt their particular behavior to just give up some part of the energy in terms of the lactate excretion. And there is one particular or extreme example in microorganism, which is they choose to evolve another way to really give up or the transported out those low energy levels. So which is a very unique, we call it, there is one bacteria called a geobacter. They can taking up the organic compounds and then use them to extract the high energy levels electron to generating ATP. But the ways they get rid of those electrons after this whole process is that they developed a filament, which is full of chromosome, cytochrome molecules on the filament, and then they becoming a conductive wires, which we call the nanowires, to the minerals, which is that contains a lot of ferric iron on their surface. And this ferric ion, it will be used as the electron acceptor for those electrons coming up from those bacteria. And then this ferric ion will be reduced into a ferrous iron. By doing this, actually this little microorganism is actually find a way to dump their electrons out into the environment. So which is very unique. And why I'm giving this very extreme example is because this is reflecting the most or ultimate purpose or important question for the cells, for the life on Earth, is that they are not only dealing with the amount of the material or nutrients they are consumed, but also they're dealing with the electrons. So they need to make sure all the electron passage and the uptaking and excrete of those electron process have to be very smooth. Otherwise, there will be a problem for life. So that's why the Albert Georgi is concluded that with a very famous sentence quote as life is nothing but an electron looking for a place to rest. So this gives us a lot of inspiration. So by doing this, as we previously mentioned, our data to find that the hoarding citrate, this behavior in those BLL cells is indicating they will be introduce a perturbation to this NADPH redox levels. So if that is the truth, that will be very interesting to us because as we know, the electron acceptor and the donor balance is very particularly important for the cell. And then when we checked this ratio of the reduced level of NADH as the electron donor, which is containing high energy levels of the electron, and also the NAD as oxidized form, which is usually work as the electron acceptor. So we found that this ratio is actually highly elevated in the double mutant, which is MYC high, KLA3 high BLL cells, no matter which type of oncogene they have to driving this malignant transformation, they both have it. And this is measured actually by biochemical methods So, and later our colleagues, Professor Zhao Yuzhen from ECUST, they actually developed a very unique tool, which is a CPYFP fused with the REX proteins, which can be used as a living cell reporter as the NAD or NADH ratio in the cell. So we use it as a reporter for our BLL cells. We have both forms. This molecule called SONA. They named it as a SONA as a reporter of the NAD, cellular NADH, NAD levels. So we can see that the regular form, the cytoplasm form of the SONA, they have a very nice localization in the cytoplasm, and it is with the mitochondria signal or location signals that will be bring the SONA cells into the mitochondria in our BLL cells. And by measuring this SONA fluorescence protein signals, we can also see the similar trend of these NADH and NAD levels in the double mutant cells, which is it's almost the same as what we observed by biochemical way. So all this data is indicating, like, of course, I'm not showing in terms of for sake of the time, I'm not showing all of them, but all the data, but I will show you the most important piece of data we have. So it is indicating that these double mutant BLL cells, they are holding the citrate, they are consuming a lot of NAD, and then they are generating high levels of NADH in the cells that could induce the cells into a state or shift this redox state of the cells from a normal into a more reductive environment, the cellular environment, which is the cells are reacting to this type of these shifted redox state by activating a lot of genes, which we call the reductive stress responsive genes in those cells. Of course, we see that the enrichment of this pathway have been observed in those double mutant cells, and also not only the cellular levels, our mouse model, but also at the patient levels, those relapsed patients or those extremely poor prognostic BLL patients, they have enriched reductive stress gene expression patterns in their tumor cells. So which is indicating all those very highly aggressive BLL cells seems to be adapting a unique metabolic behavior to making them generating more citrate, but in kind of a cost to generating too much NADH in the cell. So that's why we think it is very interesting to test if this would be a very unique vulnerability. We can really attack those aggressive malignant cells by comparing the patient-derived PDX samples and then the cell lines in our lab with the BLL in terms of the relative MYC levels and the PI3K signaling pathway. So we can plot those cells in this 2D dimension plots. You can see that the cells at these areas indicating they have a higher MYC and a higher PI3K activity. And also we measured their sensitivities to two very interesting inhibitors. One is LDHA and one is ETC complex 1 inhibitor. Both of the inhibitors are actually reducing the capacity of the cells to consuming the NADH. So which means if we push the cells into a more reductive environment, so by stopping the lactate production or complex 1 activity, they are actually generating a little bit more NADH in the cells, and it could help us to crush the overall metabolism because they're already under the stress with the reductive environment. So you can see that the dot plot or the predicted sensitivity to these inhibitors are indicating that if the cells are more on the corner, right up corner of this in terms of these PI3K and the MYC, they will be more sensitive to LDHA and the complex 1 inhibitors. And this is indicating the statistic power of this prediction, how it will be for us to predict the samples sensitivity to these two inhibitors. And also, as I previously mentioned, another cost for this citrate holding metabolism will be the cells will be kind of reducing the production of aspartate from the oxaloacetate, right? So we also measured the aspartate levels in the cells, so we can see the opposite pattern. So which means the higher the cells have this metabolic behavior driven by MYC and PI3K, the lower aspartate levels you were observed in the cell. So which we think potentially the cellular aspartate levels could be a biomarker or indicator of the cells if they are undergoing this very particular metabolic reprogramming. So we designed it as a reductive stress enhancing treatment. We called it a reset as this preclinical strategy to really testing if we can really targeting those most aggressive leukemia cells. However, we use the NUM6 and the 697 as two representative model to test later if this reset strategy would be used for to targeting them. And before that, we also would like to back up a little bit, move back to our patient samples to see if this also reflecting the similar sensitivity in the patient samples. These are samples that previously showed with the relapse and the newly diagnosis. And we found that also the relapse samples, they have the lower cellular ROS levels here by measured by flow cytometry, which is nicely recapitulating the observation we are proposing or the mechanism we are proposing. The relapse samples are in the reductive cellular environment, to which the oxidative, the opposite way, the oxidative indicator ROS will be into a lower level, so which is nicely reflecting that point. And then by measuring the sensitivity to IACS, which is a complex 1 inhibitor, we found that those relapse samples are actually more sensitive to this IACS inhibition in terms of the IC50 indicated here. So usually as we, in our general impression, the relapsed patients or relapsed samples, the tumor cells are usually very resistant to most of the chemotherapy, to drugs, right? But here, once we find exactly what is the vulnerability is for those relapsed samples, they could be more sensitive even than the newly diagnosed samples because they are becoming highly dependent on this type of the metabolic behavior. So once we're targeting directly on this vulnerability, you can kill the cells more easily. So that's we later proved that ideas in our mouse model by using the IACS as a single treatment. We see the prolonged survival of the recipient mice of the NUM6, which is the double mutant leukemia cells. And also when combined with these PEG, which is the now clinically widely used chemotherapy drug, which is actually helping the doctors to wipe out the circulating glutamine and asparagine levels in the patient's serum, actually will be further prolong the survival. We have the combinatorial benefit for those treatment for those mouse models. So which is basically what we proposed is a study we identified once these BLL cells becoming mutation or highly activated PI3K and the MYC activity, they becoming a very aggressive form of the leukemia. And then in terms of this metabolic reprogramming, we found that those cells are use a lot of glucose and glutamine through the TCA cycle, but the purpose is for generating the more citrate. And the problem by holding citrate through the TCA cycle is like you generating too much NADH, which it cannot be consumed by the ETC to generating more ATP because it's already reached the roof of this ETC capacity of those cells. And then the cells becoming more reductive in terms of NAD or NADH ratio. So if we push them further into extreme NADH high levels, you actually can very precisely targeting this part of these relapsed cells by pushing them into a reductive catastrophe, or what we call that, and the cells will be deleted or targeted, killed very uniquely. So this is what we actually found. So after doing these studies on Junzhe and Huiming and Xiaotian, they actually proposed a very interesting philosophy here. Actually, the cells or the tumor cells or other type of the cells, basically the purpose of the cell is by using these basic nutrients. For instance, we taken the example of building up a nice building, for instance, primary school here, by using a very original materials we can take from the environment. And one part of them will be generating the building blocks for the cell. And then the other part will be used up to burn in the mitochondria to generating the ATP. Both metabolism we call the oxidative catabolism and the reductive anabolism. And those two processes need to be tightly coordinated together. If there is too much generating ATP or too much anabolic activities and not matching with each other, the process will go wrong. And what we found is actually the central mechanism could be redox homeostasis is actually controlling or gearing up those two processes together to really help the cells to generating a very nice building or growth. So this mechanism also being applied by the tumor cells. And if we can find the very unique vulnerability in this central coordinating mechanism, we can targeting those tumor cells very easily. So this is what we found in my first story, which is about the B-cell malignancy. So I will probably pause a little bit to see if we have questions about my first part of the introduction. Okay, if not, maybe I would just keep going and then we can discuss later after all those data sets are shown. And the second part of my introduction or the presentation today will be, of course, about how we end understanding another type of lymphocytes, the T cells. Once we push them into the front line in the battlefield with the tumor cells, actually we now know not all the T cells will be functionally eliminating these T cells, especially if you armor them with CAR. There's a lot of CAR T cells who will undergo exhaustion or the elimination. They cannot really persist long enough to really helping the body to fight with the tumors. And what is the mechanism and what we can do to really helping enhancing this like long-term persistence of this anti-tumor T cell response. So from these studies from the T cell biologists, we now know that actually the T cells, once activated, they have to facing development differentiation trajectory. So which means some of the cells will directly derived or activated and differentiated into the effector to really counter and to really undergoing their effector functions, immune functions, and some of them will go through this TPAX differentiation and probably eventually there will be terminal exhaustion and those T cells will never be functional to really in terms of the response or for the long-term viral infection or the long-term tumor existence. And other part of the cells could be choose another way to the trajectory will becoming differentiated into a memory. And those memory T cells will somehow preserve the long-term persistence for the immune response for the next time encounter of the antigen again. And those memory cells will very fastly differentiated into the short-lived effector cells to execute their immunological function. And our previous studies actually found a very interesting metabolic checkpoint here is also in the mitochondria is also very interestingly controlling the citrate generation or consumption, which is controlled by an enzyme called IDH2. And what we found here is like if you inhibit the IDH2 using a clinically available drug called inacitinib, you can preserve more memory-like long-term CAR T cells in culture and also in vivo to helping the CAR T cells to fight with the tumors in a more long period of time. So which is the mechanisms, of course, I will not go into details, but basically by inhibiting the IDH2, you are providing the cells with more acetyl-CoA for the H3K27 acetylation, and then the whole transcriptional program will be shifting into a memory differentiation trajectory rather than exhaustion. And after finishing this studies, we are actually coming up with more questions. So as we know, the acetyl-CoA in the cytoplasm, one way will be used as the substrate for the histone modification for acetylation, right? As we previously provided as evidence. But actually, there are more other pathways that have been using up this acetyl-CoA in the cell. And then, for instance, one is will be used up for de novo lipid synthesis. And all those pathways, actually, most of the pathways have been indicated, have been studied in T cells, indicating actually some of them are very beneficial for these T cell anti-tumor T cell functions for preserving more memory-like subset. But some of the pathways, for instance, the lipid metabolism or lipid in the ways the lipid de novo biosynthesis will be performed in another way. So basically, this raised up the questions. It looks like not the acetyl-CoA levels is controlling the fate decision process of the T cells, but how the acetyl-CoA have been used is also have a contribution to the decide if the T cell will be more memory-like differentiation or goes into all the way down to the terminal exhaustion. And the one particular metabolic pathways now calls our attention will be the mevalonate MVA pathways, actually using acetyl-CoA as a very original substrate all the way down to generating the cholesterol in the cells. As we know, the cholesterol function have been studied heavily in the T cells by several different groups, including Chen Qixu from Shanghai Chinese Academy of Sciences, right? So we know that cholesterol is very important, but never seeing any single studies have been reported in the upstream activities of the MVA pathway activities have been anything to do with this T cell lineage or T cell fate decision process. So postdoc Liu Kai and Song Junzhe, the PhD student, actually they analyzed it together with the data generated by a study published on Science, which is provided by Professor Zhang Zeming. So they use a pan-cancer single-cell analysis to generating kind of a very useful data set for us to analyze. So after analyzing the T cell tumor-infiltrated T cells from different type of cancers, they found that actually the mevalonate pathway genes are actually very uniquely elevated during the process until the T cell exhausted T cells, but actually the central memory T cells have a relatively low level of the mevalonate pathway genes being expressed. So this is very interesting to us, and we would like to test it further how this pathway activity is, if this pathway activity is anything to do with this T cell differentiation process. So here is the main process. So here we know, for people already probably know these pathways, the mevalonate pathway will be named because of this generating of this intermediate metabolites will be the determining of this whole process. So will be go through down to generating the cholesterol, right? So what we down is first is to check these intermediates, the metabolite levels in T cells with human healthy donor-derived T cells in the basal level and exhausted induced exhaustion. So what we found that is after exhaustion, the T cells are actually having a higher levels of these intermediates in all these pathways. So which is quite similar what observed or predicted from the single cell sequencing data. This exhausted T cells have higher levels of the mevalonate metabolism activity. And then we would like to know what will happen if we're blocking this pathway. So we have several tools of these pharmacological inhibitors have been developed to inhibit the different process at the different steps of this pathway. And what we found a very interesting thing is that first the statin, which is stopping the generation of a mevalonate, actually can make the T cells are more memory-like but less exhausted CAR T cells during the in vitro expansion phase. And then once you're putting those T cells back into the recipient mice, they can better control the tumor growth with the CD19 as a target. So after that, we decide to putting back one by one the metabolites into the cells to see which one can fully rescue this phenomenon or this phenotype caused by statin. And we found two metabolites actually can significantly doing that is one is MBA. Of course, it makes no doubt. Like if you stop, you inhibited this enzyme pathway, but you provide more with the product this enzyme is generating, you can of course reversing this phenotype. But one thing is very important to us is like the GGPP, they call the geranyl diphosphate. These metabolites actually, compared with other type of metabolites we are providing, including the cholesterol itself, only the GGPP can rescue the effect caused by starting induce the mevalonate pathway inhibition to reverse the phenotype of those T cells. And we found very interestingly, one of the genes called PGGT1B is actually using up this GGPP as a substrate for the process called protein granulation is actually got elevated during the in the exhausted T cells. Now this experiment results help us to focus on this process is what is actually the granulation means for life. Actually, this already been studied for the as a downstream effect of these or output of the MVA pathway, not only the cholesterol production. Is the GGPP once it's have been synthesized through these metabolic pathways are being used as a substrate to be attached on a cysteine residue with a particular protein have the CAAX motif and in their C terminals. So this particular motif will be recognized by the enzyme prenylation or perennial transferase will be adding this metabolite adding on the cysteine to modify these cysteine residue and then make the proteins will be differentially modified or granule or prenylated. They were affecting the function. So one of the most important or the well-known target of this modification is the RAS proteins. After RAS proteins have been putting on this prenylation target tag on the cysteine terminals, they will be easily recruited on the membrane and to perform their to initiate the downstream signaling. So by helping the with these click chemistry, we can now label all these prenylated proteins in the cells with a Boutin based detection and technique. So here we show that these, no matter if it's a CAR T cells or just the T cell freshly isolated T cells from the healthy donors, once they are exhausted, we can see that the total prenylated protein levels, including granulation and the farnesylation, these two modifications levels are significantly higher in the exhausted T cells. So which indicating the MVA pathway mediated the final post-translational modification is got heavily elevated in exhausted T cells. And then we would like to see if this is the case by inhibiting these two enzymes mediating the last step of this modification called FTI and the GGTI, two inhibitors. And we can find that only the GGTI or the knockdown of the PGGT1B in the human CAR T cells can really enrich the memory-like subset in this during the expansion phase of these CAR T cells. And then, of course, the levels of exhaustion are significantly reduced by using the GGTI or directly knock down PGGT1B, this enzyme. So functionally, we can see that after GGTI treated with the CAR T cells in vitro and in vivo, they can better control the tumor growth with the CD19 as a target. And also in terms of these effector molecular levels got elevated from those CAR T cells treated with GGTI. And also it's got recapitulated with the genetic knockdown of this PGGT1B genes in the CAR T cells. And we performed, because as I previously mentioned, this seems to be enriched memory subset in the CAR T cells will definitely help in the mice or recipients to provide long-term protection against the tumors, right? So that's why we are using this rechallenging model in the NSG recipients using the human tumor and the human CAR T cells as the preclinical model to test this idea. Of course, we can see that after rechallenge with the second batch of the NAM6 tumor cells into the mice, the CAR T cells treated with the GGTI and the mice continued with the in vivo administration of the GGTI, showing better tumor control under the challenge with the second batch of the tumors, and then the overall survival is got prolonged by this process. So that's why we concluded that the GGTI treatment actually can prolong the CAR T cell protection upon even the second tumor challenge. So to find if this is really reflected in the real world, we find out the two publicly available data set. One is using the sequencing results for the melanoma patients treated with the PD-L1 antibody. And we can find that actually the patient T cells, if they have higher levels of this PGGT1B expression, they actually are becoming to tend more non-responders in this treatment. And the responder T cells are actually having less levels of PGGT1B. And another clinical cohort using the CD19 CAR T, sorry, there's a typo here. The CAR T therapy here is showing also the patients, the product CAR T cells, if they have a lower PGGT1B expression, actually the overall survival or the prognosis of those patients will showing better. So as I previously mentioned, it seems like this granular granulation is very important to really affecting this memory differentiation for these CAR T cells, and we would like to pinpoint which proteins is actually doing that will be most effective executioner for this process to affecting this process. So again, we use this click chemistry based labeling technique, come up with the mass spec to identify which proteins are more important. And by comparing with different data sets, we identified finally 8 candidates, which as most of them are already been reported to have been identified in other type of cells with this granulation modification. And one, that's why we have to knock down them one by one to see which one is actually playing a role in the process of the memory differentiation, and then help us to really pinpoint the one interesting protein called the PPIA. So this is showing like if you knock down PPIA, actually the cells will, the T cell will have less CD62L positive memory-like subset compared with the control. And this protein is also being identified from the mass spec with the protein particular granulation. And what is actually the PPIA? PPIA is the actually being identified very earlier in the early 90s, that being identified as a specific binding protein for cyclosporin A. And later it's been turned out to identified as the activity with the isomerase. So well, one interesting studies have been, series of studies have been done in the T cells to find actually PPIA is controlling the output of the TCR signaling through the conformational regulation of this very important kinase called ITK. So if PPIA is really interacting with this ITK, it will preventing the exposure of the SH2 domain of this ITK to SLP76 to preventing this signal transduction process all the way down. So that's why we can see that ITK here in a very central position in the signaling complex here for the T cell activation. So that's why we further to define to see if this is this true, if the PPIA is really controlling this process. We can see that after treatment with GGTI, we confirm that in the CAR T cells, the prenulated PPIA levels that got down regulated. And also from these mass spec data, we identified a very classic CAAX motif in the PPIA C terminals, and this C terminals, this motif is quite conserved from different species. So it looks like the PPIA prenylation is seems to be a very conserved regulatory mechanism in different type of the species. And then later we would like to see if the PPIA is really interacting with its non-interactor ITK. So in the CAR T cells, yes, PPIA is also interacting nicely with ITK. And once we treat the cells with the GGTI to preventing the overall prenylation, you can see the signaling output indicated by POC gamma phosphorylation from the ITK is also got diminished. So which give us the model, which is like the once the PPIA is got prenylated, it seems to be can preventing its interaction with ITK. And if this process is got inhibited, then the ITK activity will be inhibited by this PPIA interaction-based model. And then to further confirm that, we introduced the APPIA C161R mutant, which is the CAAX motif, the cysteine mutation have been introduced. That means this PPIA cannot be prenylated anymore. So that's why once we introduced this mutant form of the PPIA into the CAR T cells, you can see they have the similar effect, which is what we observed in the GGTI treatment. You have the less exhaustion of those CAR T cells and the more memory-like subset have been enriched. And to fully demonstrate this kind of mutation or this modification is really important. We using the CRISPR-Cas9 RNP technique to knock out endogenous PPIA in the CAR T cells, in the human CAR T cells, and then introduce back with the wild type or the mutant unprenylated PPRA mutant form into the CAR T cells to find out if they can what is the consequence of the interaction with ITK. Yes, you can see that once we introduced this mutant form, they have much more stronger interaction of this mutant form with the ITK here indicated. And also it's showing like this mutant form can effectively reducing the ITK signaling output as indicated by PLC gamma phosphorylation. This is done, have been done for the PPRA side, right? But how about the other side, ITK? So ITK, we introduced this P288G mutant form, which is totally disrupting the interaction of ITK with PPRA. So which means this ITK protein cannot be further inhibited by PPRA. So we can see that when introducing this ITK mutant form, which is non-PPIA interacting form, they actually have a much more stronger PLC gamma phosphorylation, and this PLC gamma phosphorylation cannot be further inhibited by the GGTI treatment. So which this piece of data to showing you like if this particular interaction have been disrupted, then the protein prenylation induced TCR signaling strengths have been also been compromised, regulation have been compromised. And by treating to look at this cellular function in killing tumor cells, we found that actually the mutant form it did not respond very well to the GGTI, and we can see that in the control CAR T cells, the GGTI can preserve more cytotoxic activity, but with this mutant ITK CAR T cells, no, they have this no such kind of effect. And then more interestingly, ITK have been identified recently as a checkpoint, a signaling checkpoint for the CAR T cell exhaustion. So there two papers have been published recently to see that ITK seems to be a nice target to inhibit, to prevent the exhaustion of CAR T cells. Of course, we recapitulated this phenomenon in our system by using the ITK inhibitor to see the similar observation. And at last, we combined with this GGTI treatment to see the most difficult field of the CAR T treatment will be the solid tumor. And by using the 143B with the osteosarcoma mouse model, we are actually, the tumor model, we are actually to showing you like this GGTI treated CAR T combined with the CAR T treatment can significantly reduce the progression of this solid tumor progression, and they prolonged the survival. And then that's why we are now presenting this scenario, which is showing you the mechanism with the T cell proximal signaling is actually controlled by the interaction between the PPIA and ITK, so which is already known. But this PPIA interaction with ITK is actually further controlled by the particular post-translational modification called granulation. Is actually controlled by the activity of mevalonate metabolic pathways. If you inhibited these pathways, you can actually provide with more memory differentiation of the therapeutic T cells and a better tumor control. So this is the second part of the stories I would like to share with you in terms of these metabolism-related protein and the signaling regulation in terms of the therapeutic T cells to helping us to generating more long-term survive or long-term or persistent T cell anti-tumor response. And the last slide I would like to emphasize is like now people talk about the immune metabolism a lot, right? If you blocking this pathway, the T cell got activated, you got memory, you got less exhaustion. But actually, it brings us a lot of, if you combine the most recently studies, especially this one is coming from the Greg Degolfi's group, is they showing, for instance, if you inhibit the very important enzymes here in the mitochondria to generating the acetyl-CoA through the glycolysis product pyruvate. If you inhibit it at the early stage or at the naive stage of the T cells, you totally disrupt the function of those T cells. They cannot be activated. They cannot exert any effector function. But if you precisely inhibit this enzyme after the activation of the T cell, you actually helping the T cells to preserve more memory-like or long-term persistent memory-like subset to preserve better anti-tumor immune response. So which I would like to emphasize here is the idea is like the metabolic reprogramming or intervention is actually can be useful only when you know when and how to be applied into a T cells during the process of anti-tumor immune response. Otherwise, it will be fully disrupt the T cell activation or the baseline or the basement of these anti-tumor immunity. So, okay, so at last, I would like to thank all the lab members in our group and all our collaborators helping us to generating this two fantastic project. And I think that will be the end of my presentation. I will be welcome any questions and discussion. Thank you.
Okay, so thank you very much, Gang. A lot of data, obviously,
Thank you.
and
Okay.
we can start the floor and then start the question. Paul, go ahead.
Hi, Gong.
Hi.
This is a fantastic talk.
Thank you.
In my life, never done the cancer research, as I always said, but I can really get what you want to express, and then the data is really impressive. I would like
Thank
to
you.
ask the second part first, very quickly. So you
Mhm.
show a lot of correlations, results, and approaches in terms of PPIA expression and PPIA downstream using different tools, genetic tools, for example, and overexpress. So why not use PPIA knocking just directly block the prenylation? Because that will be the key to me. Because you don't want to knock it out fully, right? And then you don't want to just overexpress the mutant. You
Yeah.
should just knock it in the prenylation part to directly prove that's the upstream of the downstream of the key signaling. Can you comment that very quickly?
Sure. Yes, I think that this is a fantastic idea, and we also it's our ultimate goal to really doing that in the primary human T cells.
Right.
It is very difficult for us to just generating this PPIA knockout using the Cas9 RNP system in this primary human CAR T cells, and we try to generating this knocking, as you mentioned, to introduce this particular mutant form to replacing this endogenous wild type. But we tried several times, but it's unsuccessful. So it will still be any efficient problems. We really technical problem we need to overcome to really reach that point. But we are trying. So far, we cannot provide you any positive data to showing like we can really generating this CAR T cell.
Yeah, because the GGTI inhibitor has other side effects, but I mean all the data at least support what you want to say. Another cool question.
Thank you.
It's interesting, beautiful data due to that you are aiming the metabolites of cholesterol synthesis pathway. So cholesterol, always people argue that the cholesterol can be from outside, but you are actually study the cholesterol metabolites. Just quick idea. Want to learn from you that what's a correlation of patients who take statin and the cancer in real life? Have any ideas? So in that case, the statin actually patients who take statin will have maybe correlation with tumor progression, right?
Definitely. So so far we try to search there is any publicly available data set or
Right.
cohorts you can really analyze in the starting treatment ongoing the patients and what their outcome was, especially with these T-cell related anti-tumor immune therapy, right? So that will be very interesting. So so far we did not find a very proper or useful data set we can making that statement. So that's why we would like to work with our collaborative physicians here in Zhejiang University affiliated hospital to see if we can directly uh, collecting data from their CAR T patients, because those are T cell generated CAR T will sometimes reflecting their previous, if the patient is taking starting for a long time, and their T cells to see will be helping this preserve more memory like once they have been generated into a CAR T and then infuse back into the patients to see. But it would take time. We did not so far. We did not have so many patients to enough number of patients. We can really divide them into two groups with the starting taking or now starting treatment patients to really see the difference. So I think that is a very, very important clinical
So,
relevance we would like to see.
yeah, it's a proof of principle again. If you want to
Mm-hmm.
pursue the GGPP pathway, right? Have you done, for
Mm-hmm.
example, GGPP pathway prenylation, but at the same time block the real cholesterol synthesis? Because there's a, for example, squalene synthesis, right? To really
Yes.
block the cholesterol pathway, downstream
Mm-hmm.
cholesterol production, and then
Yeah.
see if your phenotype still like holds true or.
Yes. So here I did not show, for the sake of time, I did not show a piece of data. So here is we once use this one inhibitor, ZAA, really blocking the generation of squalene from the FPP to generating the cholesterol. We see the lower the level of cholesterol in the CAR T cells by treating with the ZAA, but actually the phenotype is not as strong as what we thought, as what we expected. We did not see any side effect phenotype after this treatment with ZAA. So that's why we are focused on these GGPP pathway.
Okay, I see. Okay. Yeah, another quick question from your first part. Just want to get the first part is
Mhm.
also interesting. So I just because during the like for example the B cell clonal expansion, right? People have already showed that oxyphos is important. So different stages of